Welcome to Chapter 7, Part 1, Cellular Respiration and Fermentation for EFB103. I'm Dr. Chris Whips. Let's get started. All right, so here's a basic fundamental principle. This goes back to our unifying themes from Chapter 1. Life requires energy transfer and transformation. So different kinds of organisms do different things. So plants that can harvest sunlight energy and build chemicals, these are called autotrophs. So that means self-feeding, autotroph. Whereas organisms like you and I, we're heterotrophs. We eat different things. So we can't make our own sources of energy like plants can. But all of this, regardless of whether you're an autotroph or heterotroph, it's all about transforming energy from one form to another. Plants taking that sunlight energy, building chemicals. For us, taking chemicals and breaking those down to then build some other chemicals. So you see that as a common theme. All right, so we could look at a cellular level and connect these ideas when we think about, well, what about that breaking down? That's the process of cellular respiration. That's using organic compounds, breaking them down and extracting energy from them. That's cellular respiration. And I like this figure because it shows this connection between photosynthesis. So say inside of a plant cell or in an ecosystem even, you could think of it, plants are generating or building those compounds, capturing that sunlight energy, and then through cellular respiration, organisms are breaking them down. Plants are doing this too. So this could be inside of a plant cell, or you could separate it thinking, well, these are plants doing photosynthesis. Here's us eating the plants. So you can think of it in a lot of different ways, but all organisms use cellular respiration. All right, so quiz time. Which term describes metabolic pathways that break things down? All right, so you got catabolic pathway either through a bit of trial and error or you looked it up. Catabolism is this breaking down process. And so that's a catabolic process. That's exergonic. Think back to our previous chapter looking at free energy. And so there's different ways that organisms can do this. They can use a process called fermentation. This degrades organic compounds like sugars without oxygen, so no oxygen is needed. This is in contrast with aerobic respiration that uses oxygen and yields ATP. These other processes drive ATP production as well. Anaerobic respiration is without oxygen. So that sounds familiar. That sort of sounds similar to fermentation. But it can actually break down compounds more thoroughly. And as we'll see, what happens to these electrons that are pulled out of these compounds in aerobic respiration, they're passed to oxygen. In anaerobic respiration, they're passed off to something else. And of course, we'll get into this other part of the figure here is what is that energy used for? It's used for endergonic reactions to build things, and that's through anabolism. All right, continuing with catabolism. So cellular respiration in general includes those aerobic and anaerobic processes that I just touched on. But usually, especially at Gen Bio level, when we're saying cell respiration, we're talking about that aerobic process. And that's what we're going to spend most of our time on here. The other important thing to know is we'll use carbohydrates, specifically glucose, as our starting point, as kind of the hallmark way of working through respiration. But, remember, those other things that you eat, those can be broken down too, and they're fed in at different places along this process. We'll see this figure again later. Ultimately, all kinds of things can be broken down, including nucleic acids, which aren't shown on here. They feed in a different spot to generate energy, break down compounds to generate energy to build some more of these or other compounds. So we'll focus on the carbohydrates at first. And we can connect a couple of ideas here. What we're going to see when we talk about a sugar, so here's glucose, C6H12O6, it's going to become oxidized And because it's losing those electrons, you're going to end up with carbon dioxide. What is picking up the electrons? Oxygen. So it becomes reduced. That produces water. And then energy. And if you think about this in a different way, and that's why I show you this big picture of a fire here, think about wood burning on a fire. Remember, wood is cellulose. It's carbohydrate mostly. And as that burns, energy is released. The same thing is happening. Carbohydrates becoming oxidized. You need oxygen for a fire, right? That's a way to put out a fire, right? Starve it of its oxygen. So it needs oxygen, just like our cells do, to do this particular process. And what's released in your fire here? Carbon dioxide and water. And of course, energy. You feel that heat. There's that light energy. And so living things are doing this same basic thing, but controlling the release and preventing this dramatic release of energy all at once and slowly squeezing that energy out. Okay, so this is an exergonic process. So we see energy being produced here on the products side of our equation. And we can look back at this sort of enzyme curve that we've seen before, whether it's catalyzed or uncatalyzed. And you could apply this same looking thing to the fire burning as well, right? So you're getting the same result. You start with your reactants and energy is released. Here's that change in G, that delta G, that's the change. And then the products that are lower energy states are carbon dioxide and water, lower free energy than, say, glucose and oxygen. So remember this concept too, this idea of oxidation and reduction, and that they're coupled. So one thing is having the electrons removed from it, something else must be picking it up. So that's why we say that these are redox reactions. They go together. So what's happening here? Glucose is having electrons stripped away from it. Ultimately, they're going to end up in oxygen. And that would happen right away when we're talking about the fire example. But again, in our cells, this is done sort of a little more stepwise, and that energy is sort of squeezed out of those electrons in multiple steps. And that's how cells harvest that energy. The other thing I'll point out here is when you think about that carbon dioxide you breathe out, it's coming from the glucose originally. So here's glucose. Look what the product here from that is, as electrons are being stripped away. Oftentimes, when you're first learning about this, you think of, well, you breathe in oxygen and you breathe out carbon dioxide. Boom. Where does that oxygen part of carbon dioxide come from? Must be the oxygen. No, absolutely not. It comes from the glucose. Water is produced as a result of oxygen picking up electrons. All right, so question time. Which of the following describes the result of transferring an electron from a less electronegative atom to a more electronegative atom, which is basically what we were just seeing? Okay, so oxygen being more electronegative, it is reduced, so it gains, reduction is gain. So it picks up 2 electrons. Go back here. Oil rig, that's a handy way to remember it. Oxidation is loss, reduction is gain. And then a consequence of this is that energy is released. So that's what we're seeing here. All right, so back to this idea. Here's the fire example. So boom, we need some hydrogen and oxygen and electrons from something else, carbohydrate. Explosive release of heat and light, that's the fire example. But here's what's happening in the living thing. So we're getting electrons from carbohydrates, like we saw over here, and hydrogen, and they're passed off on this multiple step process. They're taken first by an electron carrier, NADH. That feeds them into this system called this electron transport chain. You'll see how that works. Ultimately, they do end up reducing oxygen, but along the way, that energy has been used to generate ATP. And we'll see how that's also an indirect production of ATP, which I emphasize here. ATP is synthesized indirectly. I like to think of this as taking these high energy electrons from food, and instead of just quickly dropping them down to the level of oxygen here, we go little bit by little bit by little bit, and we can use and harvest that energy. All right, so let's talk about that first electron carrier. When these electrons are stripped out of glucose, again, they don't go right to oxygen in your cells. They are passed first to an electron carrier. And one of the most important electron carriers, there's a second one as well, but this one is called NAD. And it's so nicotinamide adenine dinucleotide. And if you read that name, it makes you think, wow, that sort of sounds nucleic acid-ish. And this is something I mentioned when we talked about nucleic acids as one of these things to put in your back pocket to remember later. This is really important in cell respiration. And look at the structure of this guy, right? Here's sugar here. Got a couple of sugar groups, phosphates, two phosphates connecting them. Here's that adenine base. And then when it here's NAD+, but when it picks up a couple of electrons, it also picks up a hydrogen and it becomes NADH, all right? No longer NAD+ because it's got a couple more electrons and it's also picked up a hydrogen. And so that picking up of a hydrogen is also important to pay attention to. So what's happening when these, again, electrons are being stripped out of glucose or other organic compounds? So they're oxidations, that's a loss of electrons. Okay, so long glucose, 2 electrons taken out, but almost always you're also passing along some hydrogens, or glucose is losing hydrogens, and then an NADH picks up one of those. We'll see where that goes later. But for each electron you're stripping out of an organic compound, you're also pulling out a hydrogen, a proton. And you can see that here in this cartoon, and we could look at it in both directions, oxidation or reduction. But here's what's happening. Here's an enzyme taking this energy-rich molecule. We've just highlighted the hydrogens here kind of sticking off the side, so it's clear. So here's NAD+. And so it's ready to take those electrons from this energy-rich molecule, and it certainly does, say 2 electrons. And it's also taking one of those hydrogens. So this organic compound is now become oxidized, right? Oxidation is loss. And here goes that other proton, that leftover proton, just out into the cytoplasm or wherever we happen to be in the cell. And now here goes our NADH. Actually, I see this, it should say NADH. It's kind of, think of it as like charged up now. It's loaded with those 2 high energy electrons, and those are going to be taken somewhere else to do some work. All right, quick review. Electrons released in the breakdown of glucose end up where, given the options, ultimately? All right, this one maybe made you have to think a little bit. Where are those electrons ending up ultimately? In the fire example or in that equation that you looked at in the earlier slides? Oxygen is picking them up, right? Oxygen gets those electrons. And then so what does oxygen become? Because remember those deep protonations too, you're getting protons as well as electrons. Oxygen gets a couple of electrons and a couple of protons. What is that called? H2O, water. So that's ultimately where those electrons end up. Oxygen is the final electron acceptor, and then the product of that reaction is water. All right, so here's that full equation again. Again, we're skipping over what's happening in between, and that's really important. When those electrons are removed from glucose, those are going to be passed ultimately to oxygen, generating water, but it's what happens in between that's really important, and that's what we'll talk about next. All right, so here's the high-level overview of what happens in your cells as glucose is broken down. Energy from glucose released as electrons. Those electrons are pulled out of glucose. Electrons are transported, so first to that NAD+, which becomes NADH when it picks up those 2 electrons. And what happens to those? NADH passes them to this electron transport system. It's called that because it transports electrons. And it uses the energy, so it squeezes out the energy from those electrons. They go to a lower and lower energy state. And what does it use that energy for? It uses the energy to create a proton gradient. So think back to membrane transport and how we have differences in concentration of different things on either side of a membrane. This is active transport. It uses the energy from those electrons to drive this active process of pumping protons. All right. And then the potential energy of that gradient, that's what's used to produce produce ATP. So there are 4 stages to how that works. The glycolysis is the first step. That's starting with glucose. So glycolysis, lysis means breaking. So we're going to start with glucose and break it down into 2 molecules of pyruvate. It's almost, think of it like splitting glucose in half. There's multiple steps to how that happens. And we're going to generate a little bit of ATP from that. Then we're going to take pyruvate, the product of glucose, right? This is a very step-by-step kind of mechanistic thing. So, all right, we've got pyruvate to work on now. Now we're going to modify that and feed what's left of pyruvate into the citric acid cycle. That will ultimately completely break down glucose. Think back to that equation, right, where you end up with carbon dioxide. After the citric acid cycle, what was the 6-carbon sugar at the start of glycolysis, it's now down to just carbon dioxide. All right, where did all the energy go? It's in those electrons that have been carried away, and those feed into this last process, which is called oxidative phosphorylation. The locations matter too. So glycolysis occurs, so in a eukaryote, like in your cells, glycolysis occurs in the cytosol, but the rest of those steps occur inside of the mitochondria in your cells. Notice there's different places where we're making or generating ATP. It happens during glycolysis. You get a little ATP. You get a little ATP out of the citric acid cycle, and you get a lot of ATP out of that last step. That's really important. That oxidative phosphorylation step, that's where you get the most ATP. And so there's different ways to make ATP. But what happens in glycolysis and citric acid cycle is this. It's called substrate level phosphorylation. Sounds like a mouthful, but just break it down. You know what a substrate is. That's a thing that an enzyme acts on. And phosphorylating something is just sticking a phosphate on it. And so this is at the level of this direct building of ATP, where this enzyme taking a phosphate from this substrate and boink, sticking it onto ADP. Now you have ATP. So that's what happens during glycolysis and the citric acid cycle, very directly building ATP. The other way that ATP is built is during that last step. And I just want you to focus on this chemiosmosis part over here on the right. What we're doing, what you're seeing is this proton gradient, that potential energy, that's being used to build ATP from ADP and this phosphate pool in the cell. The phosphate doesn't come from something else that it's bound to, like we saw here, but ATP is built from those 2 components that are free within the mitochondrion. And so you can see the relative numbers here. You get a bit of ATP from these first two of these first 3 steps, but most of it comes from that last step, oxidative phosphorylation. This is also a nice figure because it shows, remember those electron carriers, we're breaking glucose down, we're stripping the electrons out of it. Where do those electrons go? They're picked up by these carriers. Some carriers are going to carry away electrons from glycolysis. They're ultimately going to end up over here. Some carry away electrons during pyruvate oxidation. Some carry away electrons after the citric acid cycle. Ultimately, they're all going to feed into this electron transport step. All right, so let's go through each one of these steps in a little more detail now. Let's start with glycolysis. Remember, glycolysis occurs in the cytosol, And we're going to start with glucose and end up with pyruvate, really 2 pyruvates per every glucose molecule. And so the things to remember when you're looking at this in a Gen Bio class is what goes in and what comes out very generally. What are the important features here? I'm going to walk through this in a bit more detail, but this slide I think sums up very nicely what you would need to know. What you see here is glucose. That's what we're going to start on. And this is the first phase of this. It's kind of in light blue here. This is called the energy investment phase. Notice we have to use 2 ATP to get this going. So you might look at that and think, wait a second, we're trying to make ATP here. Well, guess what? You do have to use some to get this going. So that's called the energy investment phase. Then, of course, here's the payoff phase. You generate 4 ATP, so you net 2. Because you invest 2, generate 4, that's your net is 2. So what goes in? Well, you also need ADP. You need some phosphates. Again, this is substrate level phosphates, so these are attacked onto ADP from something else. And you need that electron carrier, right? Because you need to carry away those electrons that are going to feed into electron transport later. So you just need a couple of those. Each one will take 2 electrons. So 2 electrons, remember hydrogens as well. And here you've generated your 2 NADH. And then what comes out? Pyruvate. Now this is the part I'll go through quickly. I think it can be helpful to look at this very step by step just to get a deeper sense of what's happening. But remember that previous slide really shows what you need to know at this level for a Gen Bio class, but this might help you learn that better. So remember that energy investment phase, boom, we've added a phosphate from an ATP. There's that phosphate attached. This gets converted slightly by another enzyme into a different carbohydrate here. And then that gets another phosphate attached. So remember that energy investment phase, there's those two phosphates. Now you have a sugar that's got the 2 phosphates attached. The next step is actually splitting that. So cleaving that into 2 3-carbon compounds, each with a phosphate attached. And then that, you can think of this actually as, for every glucose, you're gonna have 2 of these. You generate, take away a couple of electrons. So this is like a high energy compound. Let's take a couple electrons out of it. That'll slow it down a little bit. And now you have this electron carrier taking those away to the electron transport chain. And then ultimately you have this compound, here we go, producing that first couple of ATP. So there's the phosphates are taken off of this and attached to ADP. We go through another modification step. Here's that last phosphate being ripped off of these 2 PEPs, generating those last 2 ATP. Ultimately, you end up with 2 pyruvate for every glucose. And here's that summary that, again, this is the key thing, the important thing to remember, but those steps might help with that. All right, so glycolysis is done. The fate of the majority of the potential energy contained in that original glucose, where did it go? Where is it now? All right, some of you might have went with NADH because it's carrying off those electrons, and certainly some energy is being taken away with those high energy electrons. But still, most of the energy is still sitting there in pyruvate. It's, yes, we've squeezed a bit of energy out of that original glucose. Now we've got 2 pyruvate, which still have a lot to give in these next steps. So we can check off glycolysis now. We're through that. Let's go to the next step here. This is the easiest one, really. Here's pyruvate. Pretty simple, right? First of all, it gets moved into the mitochondrion. So now we're going from the cytosol into the mitochondrion. Once it's in there, carbon dioxide is clipped off. So there's the first of those carbon dioxides to be ultimately generated from each glucose molecule. Keep in mind that for every original glucose, there'd be 2 pyruvates. So if we're going with 2 one glucose, we'd have 2 pyruvates and therefore 2 carbon dioxides for every single original glucose. We'd strip out a couple of electrons here. So here's NADH picking up an electron. Again, for each pyruvate. So starting with 1 glucose, this would actually be doubled. And then what happens to what's left? This 2 carbon little nugget here. This is pretty small, and it actually needs to be attached to something else, this coenzyme A, which is this much larger compound I'm showing you down here. And what gets tacked on is, here's that little acetyl group. Those are your 2 carbons that were originally in glucose, and that's what's left of them. So they get tacked onto this much bigger thing. All right, so that's pyruvate oxidation. We're in the mitochondria now. Let's move on. Citric acid cycle, that's what's happening next. So what's going to come out of this? Well, what's going to come out, what's going to go in? Acetyl-CoA, that was from our last step. Here's the 2 carbons that were left over from the glucose. Again, if we're starting with 1 glucose, this is happening twice. You'd have 2 acetyl-CoAs. All right, so ultimately what will come out of it, you're going to finally break away those last 2 carbons. For every glucose, this would ultimately be 4. And so that glucose molecule is now gone. It's just being breathed out as carbon dioxide. And the energy from that is being stripped away and carried away as electrons by NAD+, ultimately becoming NADH. And this other electron carrier, FAD. It's very similar to NADH. It just can take somewhat lower energy electrons. We're also generating a tiny bit of ATP in this process. So that's the citric acid cycle. What goes in acetyl-CoA, you need NAD+, FAD, ADP. What comes out, carbon dioxide and electron carriers hauling these off to the electron transport chain. All right, so I'll do the same thing here, quickly go through the steps. This could be useful for some of you to understand. So here's oxaloacetate, you need that. That's the first step here in the citric acid cycle. Acetyl-CoA passes off those 2 carbons, and they're going to be colored red here, just for simplicity. The carbons in oxaloacetate are blue. And so we first make citrate. Hey, this is called the citric acid cycle because it makes citrate in this very first step. All right, so we generate some water, invest some water to make isocitrate. Next step, we take isocitrate, rip out a couple of electrons. Here goes an electron carrier, and release CO2. Now, remember I said, that original glucose will be broken down. You'll only have those 6 carbon dioxides left. The actual carbon dioxide that's being produced is from the previous cycle. So that CO2 that's colored in blue here, that's actually what is being released. Again, just remember, the 6 carbon sugar ultimately produces 6 carbon dioxide. All right, we've produced alpha-ketoglutarate, rip off another carbon dioxide, take out a couple more of electrons. You have succinyl-CoA, so you see coenzyme A making another appearance here in this cycle. And here we go. The next step, we generate some ATP, somewhat indirectly. You actually have to first generate GTP. And then we produce succinate. Now, FAD will rip out a couple of electrons from succinate, producing fumarate. And ultimately, we can take another pair. We can squeeze a little more energy out of this, and we carry away a couple more of electrons in NADH, producing oxaloacetate. And remember, that was the starting point of where acetyl-CoA feeds in its carbons. And so we've gone around the circle. Again, now for every glucose, we're going to have 2 acetyl-CoA, so this wheel goes around twice. And so we actually generate 2 NADH, etc. All right, so for each mole of glucose, or you could just think of it for each glucose molecule, it works the same, completely oxidized, so that's what's happened by the end of the citric acid cycle. How many moles or how many CO2 are released just in the citric acid cycle? So for every glucose, how many carbon dioxide did we generate just in the citric acid cycle? All right, it was 4. Remember, we already lost 2 in pyruvate oxidation, so all we really had left from those original 6 carbons were the last 4. And so that's what's released at the end of citric acid cycle. And so that glucose is gone now. It's just carbon dioxide. You're just breathing it out. We've generated a little bit of ATP, but ultimately now, where is all the energy from that glucose? It's being held in these electron carriers. Now, again, this is happening very dynamically, but we're going to look at it very step by step for simplicity. So all those high energy electrons, they're just sitting there waiting now to feed into that electron transport chain. And that's where we're going to make most of the ATP. And that's the next step. And that's where we'll start in part 2. We'll see you next time.